Welcome back, fellow scientists. So this is our last video about biotechnology. We're going to go over two processes of biotechnology. So the first process of this video, fourth process overall, is called cloning. So go ahead and write this down. Cloning is making two organisms with the same DNA. Now, this happens naturally, right? Bacteria, they reproduce asexually, so they make clones of themselves. There are some lizards, sea sponges, they reproduce asexually, so they're making clones just kind of normally, right? Humans, we actually make clones sometimes naturally. They're called identical twins. All right, you remember when we did the Plato stem cell activity and one of those little cells broke off of the morula and we said that that would become a whole new entire intact human being, a whole new person, and they would be identical twins because they came from the same fertilization event. So they have the same DNA from dad, they have the same DNA from mom. So clones happen naturally, but when most people talk about cloning, it's humans being in charge of cloning instead of cloning just happening randomly with the creation of identical twins. So here we go. This is the process. You can find this diagram on OneNote under your class notes tab. Obviously, it doesn't have the writing, but you can go ahead and fill in the writing and I'll explain that. So we take sheep number one. Sheep number one could be male. Sheep number one could be female. Really doesn't matter. We take a cell. We take any cell, any somatic cell, any diploid cell. Remember genetics talking about diploid and haploid. So regular somatic cells are diploid, one copy of the genetic code from mom, one copy of the genetic code from dad. So we have a 2N, we have a diploid cell right here. Okay. Then sheep number two has to be a female because we're taking an egg cell from sheep number two. Now remember, our egg cell is haploid, and so that only has one set of chromosomes that if it was used in a fertilization event, then would join with another set of chromosomes and then make a whole new entire intact sheep. Well, what scientists do is they remove this nucleus from this egg cell and they put it in the trash can. That's a little my version of a trash can right there. Okay, so then we take our diploid somatic cell and we use like a little electric shock and that actually fuses the plasma membranes of now this empty egg cell and this diploid cell that has a diploid nucleus in it. And all of a sudden, now we have an egg with a diploid nucleus. So that kicks things into gear. The proteins, the machinery in the egg, it recognizes, oh, cool. Well, now we have two sets of DNA. Now we don't just have one set of DNA. So now we're going to divide. And so then once the embryo gets to a certain stage, they implant it into sheep number three, into the uterus of sheep number three, which of course has to be female because it has to have a uterus. And then that develops naturally and normally. And then eventually sheep number three gives birth to sheep number four, which is the clone of which sheep? Think about it. Yeah, you're right, sheep number one, because this sheep right here only has DNA from this sheep right here, right? Sheep number two, no DNA from that because its DNA was thrown in the trash can. Sheep number three, we're just using as a surrogate mom or a foster mom right there. So this sheep number four only has DNA from sheep number one. Now, it's interesting to look at the fate of Dolly. You don't need to write this down. You don't need to memorize this, but it's just kind of cool. So normal female, during her lifetime, she gave birth to 6 of her own little lambs. Kind of interesting. She developed arthritis at about 5 years of age. Normally sheep don't develop arthritis until like 8 or 10 or something like that. She died from SPA, which is common in older sheep. So she was 7 years old when she died. Normal lifespan for a sheep is about 10 or 11. So a lot of people think it's almost as if she was born. So this is Dolly right here. It's almost as if she was born already kind of old. And in AP biology, we talk about telomeres, which are little pieces of DNA at the end of chromosomes that protect the DNA. But they, like as people, as sheep, as things age, those telomeres get shorter. Okay, so that's one idea as to why Dolly died early and had diseases at a young age that are normally associated with older sheep. So anyways, that's kind of cool. That's the process of cloning. So when we think about cloning, when we think about cloning animals, right, we always, well, okay, do we have to have human clones? Are humans going to clone themselves? Well, maybe, and you're not the way you're thinking. I don't think, at least. I don't know what you're thinking, but we have to remember back to stem cell technology when we talked about mitosis. You remember that we had this inner cell mass, and we could break that inner cell mass out of the embryo, thereby destroying the chances for the embryo to grow into a full intact human being. We could take that out and we could put it in a petri dish, and then we could expose the cells in the petri dish to different chemicals, and depending on the chemicals we could use those stem cells to make a heart, or to make a pancreas, or to make a new like meniscus or kneecap, or patella, or whatnot. So anyways, so that's stem cell technology, but here's the catch, right? This embryo right here is going to have different DNA than the person that it's going into. And so it's just going to be like this person got a heart transplant from another person. The heart's going to be more readily available because we can grow it in a petri dish, but their immune system is still going to attack it. Remember in chapter 7 where we said that the proteins on the cell membrane, that's kind of like the name tag, hello, my name is, and the immune system goes around searching for cells that don't have the right name tag, that don't have the right hello, my name is. And so these heart cells are not going to have the right name tag. They're going to have someone else's name tag. And so the immune system is going to want to attack it and reject that organ. So this person is going to have to be on immune suppression drugs for the rest of their life if they want to keep the heart. However, here we have a process called therapeutic cloning. And to my knowledge, this is not in practice. This is just theoretical, okay? So what if we take a human egg, right, out of a person, and then we take just a regular somatic cell right out of that same person. We do the same procedure, and so now all of a sudden we have a human diploid egg which could potentially develop into a full entire intact human being, which would be the same as this human being, right? But we take that out, but we don't let it develop. We take the inner cell mass out and we put it in petri dishes, and then we grow bone or muscle or nervous system tissue or something like that, and then we can use that and we can implant that back into the person that it came from. So because the new heart has this person's same DNA, now they don't have to take immune suppressant drugs, and their life, they can just kind of go on as normal. So like I said, this is entirely theoretical. I'm not aware that this is actually happening anywhere in the world right now. I could be wrong. There could be rogue scientists out there trying to clone humans and doing human cloning. But the scientific establishment has pretty much said no. No human cloning. That's a bridge too far. We can't go there. Humans are special. We can clone sheep, we can clone horses, we can clone cows and everything like that, but humans we can't clone, right? So anyways, that's just kind of one application of cloning, one special application that may be off in the future, depending on if we can get over ethical hurdles, but maybe not. It's interesting, nonetheless. It's interesting. All right, so moving on, fifth and last process is called gene therapy. So that is curing genetic diseases with genes. So up until this point in medical history, we've just been treating symptoms. Basically, I mean, yeah, giving antibiotics like killing bacteria, but for genetic diseases, we've just been treating symptoms. So there's a genetic disease called sickle cell anemia, right? And so normal red blood cells look like this. They look like, kind of indented donuts or whatnot, and they flow through capillaries and blood vessels very well. But if you have a certain mutation to the gene that makes hemoglobin, which is the main protein in your blood cells, then all of a sudden your red blood cells turn into this kind of sickle shape, and they don't flow through your blood vessels very well. They get clogged, and then they cause all sorts of troubles. Okay, so here's the idea behind gene therapy. We have a person who has no sickle cell anemia. They have a fully functioning hemoglobin gene. So we use our restriction enzymes. We cut out our hemoglobin gene. We put it into a specially genetically modified virus. Okay, and then we give the virus to a person who has sickle cell anemia, right? We give it a little bit of time to take effect, and for the DNA to find the cells in the bone marrow that make the hemoglobin, the red blood cells, and to incorporate into that. We give it a little bit of time, and then if everything works well, then all of a sudden this person who used to have sickle cell anemia does not have sickle cell anemia anymore because we fixed their genes. we didn't just treat the symptoms, we didn't just give them new red blood cells, but we fixed their cookbook that they use, that their body uses to make the hemoglobin protein. And so now they don't have to go for blood transfusions anymore. They don't have a very high likelihood of dying young. Now they can go on and they can live a normal, healthy, happy life. So this is becoming more and more prominent and more prevalent. The main thing that's difficult is that we still use viruses to transport this DNA, and so the body is still going to react the same way to viruses. It doesn't know that there's actually something good that's going to help the body in there. It just sees virus, bad, attack, and so our immune system is unknowingly working against us if we're trying to go through gene therapy. But it's really cool, it's really promising, and I can't wait to see what becomes of it. So thank you so much for watching. This has been Mr. Leo, and I will see you on the flip side.